Hi, everyone. I'm here at the San Antonio Zoo, and today we're going to be taking a behind-the-scenes tour of our Africa Live building. Behind me, we have the SSP racks, so this is where we're going to get started. If you guys tuned in a few weeks ago when I was talking at the Friedrich Aquarium, I showed you a few of the species that we're working with, and these are some of the fish that were shown there. So here we have our first species. These are the tequila split fins. These are our juveniles, so they're starting to actually mature. If you look, you can see the males actually have the little orange kind of crescent shape on the back end of their tails. The females are very bland and calm. The reason that is it's the male that has to do the work to attract the female. So he has to color up, he has to work hard, he has to look good, and then the females will pick their males. Here we have a couple more younger fry. Here. See, these guys do not have their colors yet. They're still just bear with us. Sometimes it's really hard to get through to this room, but I guarantee you the stuff I have in this room is really cool, so it's worth the wait. So, the tequila split fins, they're a type of fish that gives live birth. That means the male fertilizes the female will house the eggs in her body, they will hatch, and then she'll give birth to live young. Most other fish lay eggs. They either mouth brood like the African cichlids I'm going to show you, or they'll lay them on a leaf or a rock. They adhere them to a surface. These guys, live birth. So you might be wondering, how do I collect these young fish? Well, these are our breeding systems right here. So this is where we keep our adults. We try to keep a ratio of 4 males to every 8 females. So he's not constantly just harassing one. He has enough females to disperse his attention. So we keep our adults up here, and if you look, we have our little breeding dividers. These holes are really tiny, so when the females do give birth, their fry will actually stay under. We haven't had any offspring of these yet. They're still too young, but we still have them set up just in case. Yeah, yeah. So it's the same exact setup. If they are, you collect them here and then put them over here with more got in. Guys are all young. Over here, we have the 2 species of pupfish that we're currently working with. If you guys remember from the last time I did this talk, these guys are extinct in the wild. That means the only specimens we're checking these tanks every day to make sure that if there are any eggs laid, we are collecting them so we can hatch them. Yeah. All right. Again, I apologize. Please bear with us. This building, Africa Live, it's really hard to get signals in, but again, we do a lot of great work in here, so I do want to share this. But like I mentioned, we have the Lapoma pupfish, and then we have the Veronica. There are two different species. And again, we have these setups made to see what is more conducive to their breeding. And then here are our surplus females. We'll cycle these guys out. So once we notice a slowdown in production with the females, we'll just... All right. And this rack over here, this is our breeding rack for our Lake Victoria cichlids. These are Dagani and Melanopterus. These are the ones that we produce over 500 of each species. We have different stages of growth in these tanks. So these guys are a few weeks old. And they're very cute. And also very hungry. So Dagani, and then over here we have the Melanopterus. So again, I'm referring to my video from a few weeks ago when I talked about their breeding methods. They are mouth brooders. What that means is the female collects the eggs in her mouth, the male fertilizes, and then the female will hold the fry until they're able to free swim, which takes about 30 days. To increase our production of the fish, we actually have a really cool device, which is this. We call this an egg tumbler. So in a few minutes, I will take you to where we keep our breeding adults, and I'll explain how we collect the eggs and we put them in the tumblers.
Before we move on, we have a question from Melissa. She would like you to repeat which fish is extinct in the wild.
The pupfish. The two tanks over here, it's the Longa dorsalis, the Lapoma pupfish, and the Veronica pupfish. They're very similar in colors, but the Veronica tend to have a lot more black in their scales. The Longa dorsalis, the Lapoma, tend to have that more iridescent blue sheen to them.
Can you explain why they are no longer found in the wild, and if our goal is to reintroduce them at some point?
The reason why they're extinct in the wild is that they come from a dying ecosystem, so riverbeds are drying up. It's becoming harder and harder for these guys to stay established. There's also a risk of hybridization. There is another species of pupfish that do live in the same waters, and they're hybridizing, diluting their genetic diversity. All right. And then these guys were actually selected from our first breeding group. So these are generation, I believe, 12. So the adults are 11, these guys are 12. These guys are going to be our next generations of breeders. So we're picking our best genetic traits, and we're saving them for a rainy day. As soon as generation 11 stops producing, 12 will pick it up. And actually, fun fact, these guys up here are actually generation 13. So these are our first generation 13 babies. We do have to make sure that we don't mix the generations together to keep our program good. All right, let's go take a quick walk to my other room, right this way. So this is the room where we keep our breeding adults. Again, Melanopterus, and then we have Dagani right over here. So in the mornings when we come in, we inspect all our females. Our females are the blotchy ones. The males have the yellow bodies with a little bit of red in them too. We check them, and if we notice the female's holding eggs in her mouth, we'll actually put the date on the aquarium. And we know from that date, we come back 5 days from there, the eggs in her mouth should be fertile and good. At that time, we will actually collect the fish out of the tank with a net. We will set a container of water on top of the tank. We will grab our females and we'll put our thumb gently into her mouth and pry her mouth open, and the eggs will fall out into our container. At that point is when we set our egg tumblers. So we pop the top, we place our eggs in the tumbler, we seal it, we add the air supply, and the air supply will pull water through these little grate, keeping our eggs in motion, mimicking the environment of their mother's mouth, because when she has them, she's constantly swishing water over them, tumbling them. And with this method, we're able to increase our yield because we can pull the fish from her mouth. She will then, in about 2 weeks, she's actually able to produce another clutch of eggs. So by doing this, we can actually limit this, we can shorten the cycle between breeding events. So that's how we were able to produce as many fish as we did. And over here, I actually have more.
How you can tell if the females have eggs in their mouth. And then Jamie would like to know what would happen if we did not remove the eggs.
So we will notice underneath her jaw, there's a little pouch. And if the pouch is extended, that means she's holding on to eggs. So when we see that, that's when we tend to catch them and we strip them. If we didn't do that, then the whole process would take place. So they would breed. It would take her 30 days to grow these fish up to a free swimming size. And at time, she wouldn't be eating. She would be under a lot of stress because she would still be getting attention from the male and from the other females. So to shorten that, we tend to strip them, and we tend to get a higher yield of fish, and our females actually do better because of it. And then these are Dagonite. These are Gen 11. It's really fun working with these fish. And right now, I'm actually checking to see if anybody's holding, and everybody looks clean. So nobody's, that one could potentially be holding. Actually, that one is holding. So I will come back later, and I will probably strip that fish. And I'll probably be giving you guys an update. And then here are some more surplus Melanopterus. So in this tank, there's 97. And over here are my Dagonite. They're actually waiting to eat. So I'll give them a little treat. Whoop. It's actually empty. But in this tank, there's 497 of these Dagonite. And again, our goal is to produce as many as we can. We share these with other AZA institutions that are breeding Dagonite. And the goal is to eventually send these guys to Africa, to Tanzania, where they have holding ponds. And at that point, they'll be able to reintroduce these into the Rift Lakes. Now, if you guys have questions, don't be afraid to ask questions. I will answer them as we get them.
Nelson, can you tell us what one of your most successful stories is on fish breeding?
Well, when we took over this, when I took over this program, when I started 2 years ago, I mentioned before we only had about 20 fish. And we were able to build our SSP racks, and we were able to get these guys breeding and actually become one of the highest producer of Melanopterus and Dagonite in the U.S.
Ruby would like to know if the fish tumbler device stays in the same aquarium, or do you put it in a different one?
We take them from this system, we move them over to the breeding racks, And then we have a designated tank for tumbling eggs. So all the eggs get tumbled in one aquarium, and then as they grow, we will start putting them in the other tanks to have the different life stages of the fish. Because we wouldn't want to add baby fish in with more mature fish, there's more of a chance they're going to get picked on and eaten at that point. So, other things I could point out in this room, the best part about being an aquarist is not only do I get to work with amazing fish species from all around the world, but we get to be amateur electricians, we get to be amateur plumbers, and sometimes even carpenters. And I can actually point out some of the different parts of our system. This giant tank right here is our biotower. Its job is to provide surface area to bacteria. The two bacteria we're concerned about are Nitrosoma and Nitrobacter. They're responsible for, they convert toxic ammonia and nitrite into safe nitrate, which is nutrient, which is safe for fish. So the water goes in with ammonia and nitrite, it trickles through all this biomedia, and that bacteria is gobbling up all that ammonia and nitrite, converting it into nitrate. So it's safe, and it returns back to our systems. The other part of our life support system is the key to our life support is the sand filters. We love sand filters here. These are the most efficient way to filter water. These guys are filled to sand right about here. And what happens is the water gets pulled from the main system, goes through our pumps, goes through our multi-port, travels through our sand filter, and the water actually has to force its way down the sand. So the sand is picking up all those big particles. And the fresh water is now being pumped back into my multi-port, out, and returned to my system. So once a week, we will shut the pumps down, pop them in backwash. Basically, we're reversing the flow, so now the water's coming in through the bottom. It's agitating that sand, it's pushing that sand into this section of the sand filter that we call the effluent. All that fish waste is being knocked around. Once I pop it in my rinse, it'll flush all that stuff down the drain. I pop back into normal operation, my sand filter's clean, and my fish are healthy, and my water is polished and crystal clear. Now I can talk about one of my favorite things about one of my systems over here. This guy right here is a 400 watt UV sterilizer. This guy is amazing for sanitation. So most of you guys, I'm pretty sure you guys know about UV rays from the sun. There's UVA, there's UVB. This light actually goes into the UVC spectrum, so it goes even higher. It's great for irradiating bacteria, viruses, and protozoa. One misconception about UV sterilizer is that it kills those things, but it doesn't. What it's doing is that bacteria or viruses or pathogens are passing through the light is actually damaging their DNA. It's damaging their DNA, and because of that, they aren't able to reproduce. They actually become inert. They can still attach to a fish host, but without it being able to reproduce, it's not a threat. And eventually, it'll just fall off and die off. So, I love these things. They're really cool. These bulbs actually have the longest life of all the UV sterilizer bulbs. Most light bulbs you have in your house, they'll start to degrade over time. Most UV bulbs have about a 12-month lifespan. These guys can go up to 14 months. So every 14 months, I have to change out my bulbs, or else I'm not in that effective UVC range anymore. But yeah, these guys have been a game changer for the aquarium. We've been installing these wherever we get a chance. These things are amazing. And I prefer my stainless steel body. They cost a little bit more, but guess what? They don't melt.
Do you have a favorite aquarium here in Afterlife?
It would be HV2 Lake Tanganyika. We can go take a walk over there. Actually, I'll be honest, all cichlid tanks are my favorite. Don't tell my staff. Different behaviors. They're just a really cool fish, like our frontosas. These guys are like little gentle giants. They get big, and they're awesome. But notice their big mouths, they're mostly pescivores, so they'll eat other fish. They'll eat the small fry in here. I believe Craig talked about that last week. We have our Tropheus up here, this little brown. All right, the guys with the red fins, you'll notice their mouth structure is a little bit different, and that's because they mostly graze on algae. And again, referring to my other video when I talked about the cuckoo catfish, these are examples of the cuckoo cat. They're the ones that use the cichlids to incubate their eggs. So they do keep an eye out. If they see breeding, they'll try to sneak in there, try to put their eggs in there, and have the fish be a host. And then we have another species of fish that's actually really rare, really hard to come by, but these are the tricotti. These guys aren't normally found in the trade because they're deep water fish, so the fishermen actually have to target these guys and go very deep in the lakes to find these guys. Now, I can show you something really interesting with the hippo system. The hippo system represents Lake Malawi. Believe it or not, we're not going to be talking about Timothy. He can have his own video. And here we go. Here's a bumblebee cichlid. There's actually a female, and if you look at her jaw, you can actually see she's actually holding fry. You see how extended her jaw is? Now, I love Malawi fish, and they have a couple unique adaptations that make them super awesome. And it's all in their teeth. Let's see if I could point out some examples. Like this red one right here, this red zebra. So, yeah. So their mouth structures, you can see, if you can look in their mouth, you see they have these little teeth. They're like little rakes. Now I'm going to use my hands as an example. So this will be a rock full of algae. They have those little raking teeth. They'll swim up to the rock and they'll actually scrape and scrape and scrape, and they'll scrape little pieces of algae and they'll eat that. The other mouth structure is this guy right here, the Philoborni. His mouth is a little bit, it's kind of like a cow, very flat. So they're like the lawnmowers. So if these guys are the rakers, these guys, let me use my hands again. These guys will swim to the algae patches and munch, 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 and leave a real thin layer of algae. Now comes the pullers. The pullers, their mouth is at a 45-degree angle. And what they'll do is they'll swim up to the rocks again. They bite, they'll pivot, and they'll actually remove the algae off of the rock and eat it. So that's why they can coexist with each other, because they're all eating different parts of the algae. Some are raking, some are mowing, and some are actually pulling the algae off the rocks. And then, of course, you have the haps and the peacocks. Their main source of food is the other small fish fry. All right, and then the last lake we have represented is Lake Victoria. And this is where the Dagonite and the Melanopterus come from. These guys have a ton of color, but they're super, super aggressive. They're super mean. And me being a cichlid lover, it's really hard for me to tell the differences because a lot of the Victorian fish do look alike. But again, you can see another female right here. You can see she's holding two. So she's holding a bunch of young in her mouth. So we set up these cave systems in here so that when the females are ready, they can go in between the rocks, release their fry safely, and then rejoin their groups. But occasionally, if you look down in these rocks, you will see young fish trying to venture out. It takes them a while for them to build up their confidence, but when they do, they'll make themselves known. There's one right there. So I love these fish. They're super entertaining. They have tons of personality, and they reproduce like wildfire. If you can get them started, they'll never stop. All right, guys. Well, that's it for this section. You guys have any questions, put them in the comments. I will try to answer as many as I can. Have a great day. Bye.